Hello everyone. So as a part of this introductory neuroscience and neuroinstrumentation course, I will be next giving a short update about the wavelet analysis along with the VEPs. So what is this wavelet analysis about and what are the VEPs? A small background I will be covering in this particular slides in this presentation, followed by how to do the analysis and the wavelet analysis and everything. So firstly, I'll just give you a background about what is this VEP. VEP is nothing but the visual evoked potential. So we had the as I told you, what is an ERP? ERP is a event related potential with a particular event. How the brain is going to respond to that particular event is what is called as this event related potential. So here in this visual evoked potential, obviously there will be some visual stimulus. For example, in that P300, as I mentioned before, there was P300 for them, the 4 centimeter ball or 6 centimeter ball was being used. In case of the visual evoked potential, we will be flashing a light, or it can be either a reversing checkerboard or some visual potential is being given. So how the brain perceives this visual, it is in the visual cortex, and the visual cortex is mainly present in the occipital region of the brain. Occipital, that is in the O1, OZ, in that OZ, O1, O2, in that particular place it will be present. So here the visual potentials, it will give you it's same like the auditory evoked potential only. It will be like the having the N1, P1, P2 complex, everything. So here the experiment, it is a reversing checkerboard. The stimulus is given and it's in the frequency of about 2 hertz, which means for every 500 milliseconds there will be a reversing of the checkerboard happening over here. Similar to the other, here also we have acquired the data using the 64 channel neuroscan system. So here we have to give the stimulus accordingly. The reversing checkerboard has been given, and the artifact rejection, all the filters, the notch filter, the band pass filter, everything is being performed in this case. Basically, when there is a high contrast patterns is being observed, the brain, it will respond to that particular as a visual as a response to that visual stimulus in the visual cortex region of the brain. So how does it look like is yeah, this is how the experiment has been done. Here the reversing checkerboard is being presented over here, and the subject is made to sit in such a manner that they are in the angle is being kept to a constant, and the line of sight and all should be properly measured and everything. And then we will be the experiment was being recorded. So what happens over here is the retina, from the retina through the optical nerves, the signal is being given to the visual cortex that is present in the behind, exactly behind the brain. So it's like above the thalamus or above the cerebrum, like that. So it is at the back, and the visual potential, it is obtained, the visual evoked potential that is obtained is in this fashion. Like always in any of the ERPs, the amplitude and the latency, these two are the parameters which we have to check and everything. So for example, in MMN also we check this P1, N1, P2 response, that is the one which we check. Similarly, in for the P300 means we will check that at that 300 millisecond in that or more than that we will be getting the response. For MMN means it should be in that latency range from -150 to 300 milliseconds. So for any ERPs, any ERP analysis, this amplitude and latency is the one which we have to check in detail. So that is the one which we have to focus on in any of the ERPs. Next, I'll just review what is this wavelet about and how does that change the wavelet in the EEG analysis. So these wavelets and all, they actually give a time and frequency analysis. So frequency analysis of the EEG. Now EEG is having a huge data set, and there will be a lot of waveforms that has been obtained. So we cannot there are various methods wherein we can do the analysis of EEG, but using the Fourier transform, or it can be a short time Fourier transform, or like that. There are various methods and all are there for doing the wavelet the EEG analysis. But what happens is that in those type of channels, for those analysis like the FFTs or the STFTs and everything, what happens is there will be a loss of data, or sometimes there will be, it doesn't it should simultaneously, it has to measure, or it has to work on the time as well as the frequency domain of the EEG analysis. That has not been done in other options. Or either what happens is there will be a loss of data, or sometimes when this time domain analysis is being done, the frequency won't be done. If the frequency analysis is being done, the time analysis won't be done. So vice versa, it should be completely simultaneously, it has to be done. So only in wavelet analysis that particular option is possible, wherein we can do the analysis simultaneously, and the correlation studies and everything can be done together. So what is our wavelet all about? Wavelets are nothing but with the whole of this our EEG data, we have to decompose it into small wavelet packages. So first of all, based on this particular step, it always takes place based on the sampling rate alone. So with the sampling rate only we can do the wavelet analysis properly. Based on that only the different wavelet coefficients are being created. So here in this analysis we have we have been working with the wavelet analysis that has been done by Quiroga. This Quiroga's paper, they have made a software, or there is something called as the EPDEN software, which has been given in the University of Liechtenstein. So in that particular, if you just put EPDEN wavelet denoising Quiroga, if you put in this way, you will get in Google you can get their page and how to download and everything. So it's like they have their MATLAB code itself. So it just we have to run it, and we have to give the data according to that. So another one important thing about this wavelet is that it always takes the sample number or sampling rate or anything in the powers of 2. That is one of the most important thing, because that is like a standard. It should have always a sample size, it should be in the powers of 2 alone. Okay, so the sampling rate over here which we take, it is 256 hertz. That is a normal, that is like they have used for their tutorial purposes, their analysis, they have took 256. So according to our data, according to our CNT files, we can give this particular sampling rate according to that. So for example, now in all the neuroscan data files, the sampling rate has been taken as 1000 hertz. So what we do is that we actually will resample those data in 1024, which is again the power of 2. So that is the important step we have to consider. Now in case of Inobio or the open BCI data and all, they take a sampling rate of 500 hertz. So in that case we have to do a closest to the powers of 2, means that will be 512. So we have to do the resampling in that 512 range. So based on this sampling rate, the wavelet coefficients will be decomposed. So basically what we are doing is that the whole data set, first we do the whole data set is first run by this data wavelet coefficients, then we reconstruct the data, and then we get the wavelet analyzed after denoising with various algorithms and all. So in this particular Quiroga paper, they have mentioned that they have used something called as the B-spline orthogonal they have used that particular wavelet. So that is the that is like that is is somewhat similar to how the ERPs look. So that's why that particular orthogonal B splines are being used. These wavelet coefficients, they will be divided, or they will be as I told you, they will reconstruct, I said, no. So first we have to, they will first reduce it in this D1, D2, D3, D4, D5. So in this ranges, the gamma, beta, alpha, theta, delta, in this ranges, it will be in this particular range, it will first get reduced, and then from here it's been reconstructed as the next for the next wavelet for the analysis. So first what happens is, after doing this EEG analysis and everything, the data is then exported. I was showing how we export the data. So after exporting the data, we have to convert those datas in the forms of we have to convert it into ASCII files, and that too it should be all those sample data also, it should be in the powers of 2. I'll just go through the wavelet analysis, the software, the code, and I'll show you where the difference and all has to be made, and a single column ASCII file has to be made. So these are the next 3 additional steps that has to be done apart from the EEG analysis, ERP analysis. So I'll just do this extra step to show how to do the wavelet analysis and where the changes and all to be done in the code. So this is just a sequential step of what is the demonstration, what I am going to do. So similarly, we first do since we are working on the VEP data, first we will do the VEP analysis, and we have to do here one important step we have to do is the resampling we have to do, because we want to get the samples, export the samples in the powers of 2. So for that purpose we have to do the wavelet analysis. So this resampling has to be done, and then followed by the similar thing, epoching, the creating the event list, then the VEP triggers, then all those VEP analysis, everything has to be done, and finally we do the wavelet analysis using the software. So this is how we do the this is the just demonstration steps that I'm going to do. Next I will be showing how to do the thing. Before that, just these are the 2 Quiroga papers I have mentioned about the wavelet analysis they have been doing. So in their website also there will be various tutorials and everything that shows how it looks when the data things happen. And the wavelet analysis, important thing is that we can also it also helps us to understand how our brain is getting used to the stimulus. Now for example, an experiment, it takes for about 15 to 20 minutes, means initially for some time our brain will be a bit it will get used to I mean it will be like first initially we will be concentrating on the triggers or on the stimuli, everything, but later on we will be our brain will get used to that. So that habituation, it will take place in our brain itself. So that also can be analyzed using the wavelet analysis, how the habituation is taking place as the time proceeds in the experiment. So based on that we can also tell, okay, this is how the brain should look. I mean this is so much time is required for obtaining the P300 analysis or AEP analysis or VEP analysis, that we can the time required for the thing can be understood. And this is the first 2 papers are just some VEP related papers wherein they have used the VEPs, how when and all that has been when they have started working on it and how it was being developed, and everything has been shown in those 2 papers. Next I will show you the demonstration. Thank you.